All right, let's talk about a virus that's on the World Health Organization's priority list, one with known pandemic potential, but I'm willing to bet a lot of us have barely even heard of it. It's called Nipah virus, and to really understand the threat, we have to start with one single kind of terrifying number.
In some of its outbreaks, the case fatality rate for Nipah has hit 80%. No, that is not a typo. For every 10 people who get infected, as many as 8 of them have died. We are talking about an incredibly lethal pathogen.
So you can start to see why this is such a big deal, right? When you have that kind of lethality, plus its ability to jump from animals to people, and evidence that it can spread from human to human, well, you can see why the experts are so concerned. We're going to break down exactly what makes Nipah tick and how a really brilliant piece of science unlocked one of its biggest secrets. To really get a handle on the risk, we have to look at where Nipah has been and how it actually operates. This isn't some brand new virus, and it's definitely not stuck in one single place. This threat has been building for decades now. It first popped up in Malaysia way back in 1998, jumping from pigs to farmers. Then, in 2001, it showed up in Bangladesh, where it has caused outbreaks almost every year since, this time linked to something as simple as contaminated date palm sap. We've also seen it in the Philippines, jumping from horses, and in India, where recurring outbreaks in Kerala are a huge public health challenge. Every single outbreak seems to show us a new side of this virus's adaptability. So how is it making all these jumps? Well, its natural home, its reservoir in nature, is the Therapeutic fruit bat. The virus just hangs out in them, totally harmless. But then it spills over. It can go from the bats to other animals like pigs or horses, and then to us. It can go directly from bats to humans, a lot of times through things like raw date palm sap that's been contaminated. And this is the most worrying part. It can spread from person to person through close contact. And that brings us to a massive problem. If you want to fight a virus, you have to be able to study it. But how do you study one of the most dangerous pathogens on the planet? The answer is very, very carefully and with a whole lot of difficulty. You see, Nipah is what's called a biosafety level 4, or BSL-4 pathogen. That's the absolute highest level of biocontainment there is. Think of it like a maximum security prison for viruses, where scientists have to work in these pressurized spacesuits. This level is reserved for the worst of the worst, like Ebola and Nipah, viruses that have no vaccines or cures. And this quote from a really important research paper back in 2004 just nails the problem. Working in a BSL-4 lab is slow, it's expensive, and it is incredibly restrictive. There are only a handful of these labs in the entire world. So that reality just severely limits how much research you can actually do on the live virus. So this creates a fascinating puzzle, right? How can you possibly study the virus's rulebook if you can't even safely open the book? To figure out its fundamental operating manual, scientists had to get really, really creative. And that is exactly what they did. This brings us to the absolute core of this whole story, the Rule of 6 breakthrough. It was a groundbreaking experiment that figured out a way to understand the virus without ever having to touch the live, infectious thing. The solution was just brilliant. They built a safe, harmless viral decoy. It's a system called a minigenome assay. Basically, instead of using the whole deadly virus, they just used a few of its key parts in a totally safe, normal lab environment to watch how it copies itself. Okay, let's walk through how this works, because it's really elegant. First, they took the genes for just 3 viral proteins that are absolutely essential for making more virus. They're called N, P, and L. Second, they built a tiny, harmless little piece of the Nipah genome. Third, and this is the clever part, they stuck a reporter gene into this mini genome. Its only job was to light up or produce a protein they could measure, but only if replication was actually happening. And finally, they just put all these pieces together inside safe lab cells and waited to see if they got a signal. And this is the eureka moment. They tested these little mini genomes with slightly different lengths. When the length was 875 units, or nucleotides, nothing. When it was 877, still nothing. But when the length was exactly 876, a number that's perfectly divisible by 6, boom, they saw a massive spike in that reporter protein. Replication was working, but only when the genome length followed this one very specific mathematical rule. This amazing discovery confirmed that Nipah virus follows what's called the Rule of 6. And this isn't just some weird quirk. It's a fundamental part of its design. The virus's N protein, which basically acts like a protective coat for its genetic material, grabs onto that material in chunks of exactly 6 units. If the total length of the genome isn't a perfect multiple of 6, the whole replication machine just grinds to a halt.
So then the researchers took this system and used it to test other viruses. When they took the Nipah replication machinery and tried it with a mini-genome from its super close relative, Hendra virus, it worked perfectly. But when they tried it with a more distant cousin, the measles virus, nothing. It failed completely. It was almost like a genetic paternity test, and it confirmed that Nipah and Hendra are in their own special family, called the Henipah viruses. So let's zoom back out and connect this incredible piece of basic science to the bigger picture. I mean, why does knowing about a rule of 6 actually matter in the real world when you're in a race against a deadly virus?
It matters because every rule reveals a potential weakness. Understanding this fundamental assembly instruction gives scientists a very precise target. If you can design a drug that messes up this perfect 6-step process, you could stop the virus from ever being able to copy itself. Discovering the rulebook is always the first step to writing a battle plan. And we desperately need that battle plan because the reality of where we are right now is, well, it's pretty stark. Nipah is still a WHO priority pathogen. We know it has pandemic potential. And right now, today, there are no approved vaccines and no specific drugs to treat it. All doctors can do is provide supportive care. So that leaves us with this massive question, doesn't it? How do we turn this kind of incredible, fundamental research into actual global readiness? With the threat of new outbreaks always lurking, the real challenge is figuring out how to speed up that journey from a foundational discovery, like the rule of 6, to the vaccines and treatments we need to actually be prepared.